Good morning. Today we are now on the week 2 of Quantitative Genetics, and what we are going to do now today is to look at how we are going to calculate for the population, mean, and so on. So let's go to the first slide. I spoke to you about this, and as you said that characters are given a value and they're expressed in metric unit, and phenotypic values are characters that measure on the individual. Phenotypic value equals the genotypic value plus environment. So I am reaffirming what I said just now. P equals to G plus E. P is the phenotype, G is the sum of genetic effects, and E is the sum of all environmental factors. So the assumption here is the environment deviation in this population is 0, and in this case P equal to G, and the population mean will equal to genotypic value. We assume that over the generations it's constant value. Now let's talk about dominance, codominance, and overdominance. Consider a single locus with two alleles, A1 and A2. We give the genotypic value of one homozygote plus A. That means A1 is plus A. And for A2 we give it as negative A. This is just a value we give assuming that A1 gives a positive value and A2 give a negative value. And the heterozygote we give a value of D. This is a scale. So when we talk about metric characters and values, we give them a scale. So if D equals to 0 is when there's no dominance. D is positive when A1 is dominant over A2. D is negative when A2 is dominant over A1. D is either positive A or negative A when the dominance is complete. If the D is above than positive A and less than negative A, there is overdominance. Now the degree of dominance is expressed as D over A. This is very important because in quantitative genetics you are supposed to quantify everything in metric values. So you need to understand this concept for the rest of the course. Let's make sure that you understand more. We'll give you an example of the pygmy mice. Pygmy mice is now being heavily investigated because they're known for their cuteness, and they're also known as a dwarf or small mice. So pygmy mouse is known as a dwarf gene, noted as PG. It was discovered. The mutation was discovered in 1950 by Professor MacArthur's. Okay, and in collaboration with Dr. Falconer, they wanted to prove the inheritance pattern in the mice when they crossed the C57 strain and small mice strain to postulate the hypothesis. Let me share their observation. So in the weight of mice at 6 weeks of age, this is taken from Falconer 2008. These were the genotypes of the mice. So if it has the pygmy gene, which is the dwarf gene, the weight is around 6 grams. If the heterozygote was the normal and the pygmy gene, the weight is 12 grams. And if it doesn't have the pygmy genes, the weight of the mice is 14 grams. Now, how do we quantify the values in form of metrics? You can see that if you find the midpoint genotypic value in this population is actually 14 plus 6 is 20. 20 divided by 2 is 10 grams. So if you look at the value of A, the value of A is actually 4 gram and the value of D is 2 gram. That's why you see the results as such. I hope you understand with this very simple example.